Evidence of evolution. Searching for common ancestry. Brought to you by Curious Moranland, where science literacy will make America great. So, if you look at this list, you might get a variety of different lists on this, and a lot of people now are starting to combine these two, but I'll elaborate on those. So, I'm not going to talk that long. I just want to do a quick overview of things that I did in my classroom. I have a separate PowerPoint and things that go with fossil evidence, so I'm not going to talk much about them. But they're one of the things on the list. But if you look back, I didn't say comparative fossils, because you can study comparative anatomy and study fossils, or embryology if you're fortunate, and various other ones. So I'm going to talk more about comparative anatomy. I'm going to do a separate video about biochemistry and finding the link between chimpanzees and humans, with chromosome 2 for us and 12 and 13 for chimpanzees when they fused. Okay, so comparative anatomy. So we're looking at three basic terms when you're looking at comparative anatomy. Structural similarities link the species. Structural similarities are going to link the species. So let's usually I jump right to here. This is a set of notes that my students copy. And the thing to see from this is different ancestors, but they end up having similar functions. I'll show some pictures in a second. Different ancestors, similar functions. That the evolution there has a different angle versus similar ancestors, but different structures, different functions of those structures. So different environments. So let's look at. These are all from mammals. And one of the things you'll notice is they've got a single bone that's going to attach to the body, a single bone, a single bone, a single bone, followed by two bones, two bones, two bones, and then a smaller set of bones, which we'll call carpals, and then the metacarpals, and then the digits, or the phalanges. Look at the phalanges here. Each one of these serves a different function. We don't walk on our hands, not regularly, unless we're a gymnast, but we don't walk on our hands, but we have this opposable thumb. We're going to grasp things. These other three, it's for transportation. So, analogous structures, look at the 5 structures here. One of them is vastly different in its biochemical makeup and underlying structures than the others. However, it has a very similar function. So that's when we're going to say it's a analogous structure. Same function, different, similar environments, but they are not more closely related, unless you're going back to the pre-vertebrates. Okay. Now, if you look at these, now let's do a comparison of homologous structures. Eliminate the wing for the fly. And now, the arrangement of these bones are going to have a similar function. The bird and the porpoise. All right? The bird and the porpoise. However, the bird and the porpoise, although they have a similar function, one for transporting it through water and one through air, the bird is the least alike of the 4 homologous structures. Porpoise, mammal. Elephant, mammal. Human, mammal. Bird, aves. So what we do is we compare, we group on the basis of similarities, the homologous structures. We separate on the basis of differences. That's basic classification and taxonomy terms. So let's move down to another structure, vestigial structures. These are structures that no longer have a function. The problem that I see students is they say, oh, well, they didn't need it, so they didn't use it. That's a Lamarckian explanation, and we're not accepting Lamarck's explanation. He helped us by understanding that organisms change, but we accept Darwin's descent with modification and the driving force of natural selection. So vestigial structures, they were common, but they are no longer being used. There's a term called atavistic genes, which I think are going to become more widely used in the science world and genetics and evolutionary studies, and I'll refer to those a little bit later on. But these are basically genes turning on and off. And now we'll bring in the link between natural selection shifting a phenotype to being expressed versus shifting a phenotype to not be expressed. So that is one of the newer ways of describing it. So just to show you vestigial structures, a whale has the hip bones for a swivel type motion, but they use their bones for gliding through the water, transporting themselves through the water. The scientists are debating about whether the appendix is actually vestigial or not. We know it helps with vitamin D metabolism, and it helps when we have a more heavy cellulose-based diet. So comparative embryology, what we're looking at is similar embryology, similar development, more closely related. And I really didn't say that well. So let's look at this. The embryology here, these are all mammals. These two are mammals. This is a chicken, this is a fish, but these are all vertebrates. That's what I was trying to say. And although we don't have gill slits now, but when we are developing, we start with them. Embryological development, the studying of fetus. From that fertilized egg, the embryo starts to divide and then specializes, and when we're calling it a fetus, it starts specializing in the different tissue. For example, this is just restating what I've shown, but some of our cells are going to, the webbing we have here are going to start breaking down in that webbing. Lysosomes, the cell structures, are going to increase and start dissolving these substances. Genes turn those on during embryonic development. I'm predicting that the field of epigenetics is going to revolutionize embryological studies and genetic genomics. So I'm going to finish up with the next two. If you're one of my students and you haven't had a chance to copy these down, I would pause the video and copy this down, or go back to the beginning and pause as you're going along. I should have suggested that earlier. This looks very similar. The only difference between these two slides is one we're saying we're comparing amino acids, one we're comparing DNA. So let's look at comparing amino acids. At the top we see a human. This is the amino acid sequence for protein. A protein that we call hemoglobin. It's a red blood. It's in our red blood cells. So if you look at the chimpanzee, there's really no difference. Look at the gorilla. One difference. Now the least related would be the frog. It has so many more differences, which makes sense that mammals would have less differences. So there's varying levels. You can separate out gorilla down to the rest of them by the one difference. Chimpanzee and a human, there's no difference in our amino acid sequence. I'm going to do a video later where we talk about the chimpanzee and human similarity. It's only about a 1 to 1.6% difference. And that 1% difference is amazing. We'll look at that later. So I have over here this phrase genomics. All right, we're talking about understanding with the Human Genome Project. We've taken DNA and amino acids and RNA and understood that if you know the DNA, you can go back to the RNA. It's kind of like reverse transcription or reverse translation. If you have the amino acids, you can go back to the RNA. If you have the RNA, the mRNA, you can go reverse transcription and go back to the DNA. It's those fields all merged, and this is going to relate some really great findings as in the near future, and we're doing it right now as well. So I'm going to close the video with genetic evidence, mitochondrial DNA. Genetics is revolutionizing the study of evolutionary biology, and the old days where you say it looks like, therefore it must be alike. We've long since learned that that's not correct. But we're not going to throw out studying comparative anatomy, but we're bringing in all this. Each one of these studies is further evidence confirming what the older studies did to now, and that is organisms that are more closely related have common ancestry, and we use different methods to do that. Thanks for watching. Brought to you by Curious Moranland.